Hello friends, in the first part of traditional food preservation technologies, we studied what are the different methods which are traditionally used for preservation of food, what are the target factors in these methods, what is the objective and the mode by which these objectives are achieved. We saw that there are three sets, one sets of technology are used to slow down microbial growth, other sets of technologies are used to kill or inactivate microbial growth, and other technologies are used to reduce or lower down the contamination or restrict the access of microorganism. In all these processes, three major things we saw, that the control of water activity, control of pH and heat treatment are the major factors which are used to preserve the food traditionally. We discussed little details of these aspects and also saw that the heat treatment, in fact, how when we give a heat treatment to a food, the shelf life of food is increased, and in fact, thermally processed food constitute a large part of the food processing industry or the products which are available in the market. So, let us study in this part of the lecture or today, that is traditional food preservation technologies part 2, we will basically discuss, that is what happens, how the microorganisms get killed when the heat is given or heat energy is given to the food. The impact of heat treatment on microorganisms is estimated from the measurement of the surviving cells against the heating time. The destruction profile that is obtained allows the determination of the characteristics of the resistance of the microorganism, and that are the basis of the designation of the operational requirement of the thermal process. That is, what should be the time temperature combination of a thermal process. This is mostly based upon the total energy input that is required for the inactivation of most heat resistant form of the generally spore of the bacteria. Traditional estimations of the efficiency of preservation and disinfection process are based upon the assumption that microbial death follows a known evolution. Each species of the microorganism has its own particular heat tolerance. Thermal operating conditions are determined experimentally, and the data are presented as the number of surviving microorganisms or viable spores against the exposure time at a given temperature. That is, different experiments under different temperature and time conditions are conducted, and the effect of these time and temperature combinations on either killing or survival of the microorganisms are estimated or recorded, and these data are analyzed to find out that how the changing in the temperature is causing or influencing the microorganisms and their survival in the heat process, and these are basically used for designing of a heat process. So, the thermal destruction kinetics of microorganism become an important subject in the thermal process design. So, we will see that is thermal death kinetics or survival curve. That is one of the major thing is there is to these data which are generated experimentally on the most heat resistant vegetative cell or spore of the bacteria. They are plotted in the form of a survivor curve. That is when the log number of surviving bacteria is plotted against a time, generally a straight line is obtained. That is log number of survivors versus time, we get a straight line. And in as you could see, that is the line 1 in this figure, that in many cases this representation gives a linear relationship, and this linear relationship implies that thermal destruction phenomena is a first order reaction. However, as you could see in the figure 2 and 3, the deviation from the thermal death curves may be observed, and this thermal death curve can also be characterized by soldiers and trials. The deviation from the linearity may be related to the composition of the microbial population, germination of sporulated forms, or heterogeneity of the thermal treatment. In this graph, again we are showing the 3 lines, that is, it is a schematic representation of the 3 types of microbial survivor curves, representing the number of viable cell against the time, that is log number of survivor against the time. This line 1, which is a linear relationship, it gives a linear relationship straight line. This curve shows that in the population there is a homogeneous mass of microorganism, that is it is a homogeneous population of microorganism, and it has been homogeneously heat treated. That is all the microorganisms, they have got similar types of heat treatment. The line 2, it is a typical curve of where composite population, that is thermosensitive as well as thermoresistant cells coexist in the system. The third line is the curve of microorganisms that are activated by short exposure to heat, like when the heat is given to the system, some of the spores might germinate, or there might be certain fragmentation of chains of the microbial chains, and their number. So, this means that is the line 2, so that the coexistence of strains having different thermal resistance gives the segmented lines. Thermal activation of germination is responsible for maintaining the initial cell number, which is shown in line 3. So, in the straight path of these plot, whether plant 1 which is fully straight, in the 3 and 2 also there are some portions which are straight. So, in the straight part of these plots, the number of cells thermally destroyed at temperature T during the time interval delta T seconds is represented by the equation x t plus delta t minus x t is equal to minus k x t delta t, where x is the number of living cells and k is the destruction rate per second. Equation 1 can also be written as x is equal to x 0 e to the power minus k t it is exponential form of equation 1, where x0 is the initial number of microorganism, that is the cells initially present, x is the number of survivors after the heat process, t is the time of exposure in seconds, and of course, k is the destruction rate constant. And in this, that is k in the microbial death process, a concept of d value, where that is the log of numbers of survivors is plotted against the time, right, or there is when it is plotted on a semi log cycle, that is if you say number of survivor, it plotted against time in a semi log curve, 0, 1, 10, 100, that is 10, 10 to the power 2, 10 to the power 3, 10 to the power 4. So, this gives a straight line, and in this line, that is the time required to reduce the microbial population by a factor of 10 is known as d value. That is the, it is called decimal reduction time. That is the average time of exposure needed to reduce the number of microorganisms by a factor of 10. And if we use this d instead of k in the equation 2, it becomes x is equal to x0 10 to the power minus 1 by d. Another parameter which is used to indicate our thermal resistance of microorganism is Z value. So, like d value, where log number of survivors were plotted against time. So, if d value is plotted against temperature, that is on a semi log cycle, 1, 10, 10 to the power 2, 10 to the power 3 and so on. So, we get a straight line, and the slope of this curve is Z value. That is, Z value, it corresponds to the increase in the temperature that induces a reduction of d value by a factor of 10, and this is given by a equation, that is d is equal to d reference, it is 10 to the power minus t reference by Z, where T is the in degree centigrade is the temperature of the treatment, D reference is the thermal reduction time at the reference temperature, that is T reference, and this reference temperature for pasteurization process is generally 71.7 degrees Celsius, for the sterilization process it is 121 degrees Celsius. And this Z value in fact is used, we can conduct the experiment, we can find out G value at different D value and different temperature, and then this D value of different temperature can be plotted and Z value might be found, and this Z value gives a good indication about the thermal resistance of the microorganism. That is, let us take two microorganisms are there. One microorganism has a Z value of 10 degree, another microorganism has a Z value of 50 degree. Then the microorganism which has a higher Z value or food component or reaction which has a higher Z value, that will be least influenced by the temperature. In other words, that is, one has to increase the temperature 50 degree to get that 1 lakh cycle reduction in D value, whereas in other case one has only just by increasing the temperature 10 degree we can get a 1 lakh cycle reduction in D value. So, the components that have a small Z value are highly temperature dependent, they can be more influenced by the temperature. So, the thermal process calculations therefore involves the need for knowledge of the concentration of the microorganisms to be destroyed, the acceptable concentration of microorganisms that can remain in the final product. Okay, we have to decide whether we want to go for 12 lakh cycle reduction or 8 lakh cycle reduction or 5 lakh cycle reduction, depending upon the number of or type of the microorganisms and food characteristics, etc. Also, the thermal resistance of the target microorganisms and the time temperature relationship required for the destruction of the target microorganisms. There are specific D and Z values for the thermal inactivation and destruction of enzymes, vitamins and pigment, etc. That is enzymes, vitamins and pigment, other matter the food constituents and the microorganisms, that is the unwanted elements in the food like pathogenic cells, toxin and enzymes, or desirable components in the food like nutrients, flavor and color. They have different D value as well as Z values, and these differences of the D values and Z values of these undesirable and desirable components in the foods are generally exploited to optimize the thermal treatments, or they form the main basis for the optimization of the thermal process conditions. Here we see one, that is a relative change in the time temperature profile for inactivation of microorganisms and the destruction of vitamins in a food. We see in this figure. You can see as I told you earlier also, that Z value and D value for these components differ or vary. For example, Z values of around 10 degrees Celsius are the characteristics of the pathogenic bacteria, whereas Z values for enzymes, vitamins and pigments generally are found in the range of 20 to 70 degrees Celsius. Okay. And this difference allows the manufacturer to determine an optimal thermal process condition like temperature and exposure time, combination of temperature and exposure time. And in fact, in the things which is shown here, it actually forms the basis of high temperature short time treatment, because most of the thermal processes, these thermal processes may be of low temperature long time or high temperature short time. The low temperature long time methods generally lead to the destruction of the nutrient, and high temperature short time, because the temperature exposure of the components, food component to the temperature is very short. So, it results into the better nutrient retention, while the desired microbial or undesirable components destruction. You can see here in this graph that this is a line, all right, which shows the inactivation rate for that, or curve is this curve for the pathogens. So, on this side, that is the pathogens, normally it is the log D value has been plotted against temperature. So, means that is Z value, which are the normally pathogens, they have Z value less than 15 degree, all right. It is Z value 10 degree, but we are taking that less than 15 degree. So, on this side of this line, they will remain intact, whereas the food nutrients like vitamins, color, flavor, etc., they have a higher Z value. So, here that is Z less than 30. So, 15 to 30 we taken this line. So, in this line is we see that is these vitamins will remain intact, or above this if you go, the vitamins will get destroyed. In this line, above that line you go in this region, the pathogens will get inactivated, right. So, if you see in this place here, in fact, here although this pathogens will remain there, or vitamins may get more destroyed. So, it is the shadowed line here you are getting, that is the pathogen also get inactivated, as well as the food, that is desirable constituents of the food, vitamins, pigments, colors, etc., they are also retained. So, it represents the region where the thermal parameters allow preservation of vitamins and destruction of microorganisms, and this gives that high temperature short time treatment. Okay. So, this that is it gives the curves of pathogenic organism, microorganism and vitamins. Now, like the growth and multiplication of the microorganism is influenced by certain factors, similarly that even inactivation, destruction of the microorganism is also influenced by certain factors, like the classical microbial inactivation models based on the temperature levels and exposure times are highly influenced by intrinsic and environmental conditions during the thermal treatment. That is what are the conditions prevailing in the system in which the heat process is given, they also influence the thermal destruction kinetics of the microorganisms. So, these factors also should be considered, and one important factor this is heating rate, the rate at which heat process is applied, and which generally not taken into account in the models of thermal destruction. Although it has been found to be a determinant parameter affecting the survival of the microorganisms to a heat process. Even the heat shocks are found more effective than the progressive heating, and it enables the heat optimization of thermal destruction process by increasing that heat gradient. It means that instead of giving a continuous, if we give heat shock, it has more influence on the microbial death or microbial death rate. At a fundamental level, the effects of heating rates on the membrane permeability also emphasize the fact that cell membranes are probably structurally dependent on heating kinetics. The other factor, that is the environment in which the food is heated. The also environmental parameter like water activity and our hydrostatic pressure, they significantly influence thermal treatment effects on the viability of microorganisms. Microorganisms appear to be thermally stabilized by intermediate AW water activity values, that is in the range of 0.3 to 0.5, and the most favorable hydration conditions for their destruction correspond to fully hydrated or dehydrated media. As proteins are generally stabilized by extreme dehydration, it can be speculated that the thermal destruction of cells in dehydrated media is enhanced by the oxidation or by membrane destabilization favored by low hydration. The major role played by membrane destabilization in the production of cell death can be seen in the way that passing through the membrane phase transition using a cooling step enhances cell resistance to osmotic dehydration significantly. The main implication of these results is that thermal stabilization of dehydrated products can be optimized, especially when high heating rates are used. The combination parameters, they also, that is the combination of parameters also influence the heating process or thermal destruction of microorganisms in food. The association of hydrostatic pressure with temperature is an efficient means to control microbial survival. One very interesting aspect of this interaction, that is interaction between hydrostatic pressure and temperature, comes from the elliptical shape of the viability curve and temperature pressure diagram if we see. And these curves, which are generally analogous to the protein denaturation curves, suggest that the inactivation effect that are obtained by heating under pressure can be more efficiently obtained by cooling at negative temperature under pressure. And in fact, this forms the principle of that hydrostatic pressure, hydrostatic high pressure processing, that is the cold temperature high pressure inactivation effects is usually a promising mean to provide safe, but non-thermally affected food products. And this forms the principle of the high pressure processing of the food, which is one of the emerging technologies in food processing now, and this we will take up separately in one other lecture. The water activity and hydrostatic pressure are two thermodynamic parameters. They are rarely taken into account in thermal process calculation models, okay, or the models of thermal inactivation, but that greatly modify the thermal effects observed in classical condition of hydration and pressure. So, hydration and hydrostatic pressure levels as well as heating rates strongly influence thermal stabilization of food products, and these parameters could be more efficiently used to improve classical heating process and therefore, to get better effect of the process control. So, by proper control of these parameters, that is if we understand that is how changes in the heating rate, changes in the or by having a proper combination of the heating process as well as the environmental conditions, that is during which the heat is given, having a better heat transfer efficiency, we can control, we can have a better, that is thermal process, which will result into a desired sterility level in the product. At the same time, it will maintain the desired level of nutrients and other quality factors in the food. So, this thermal process or thermal destruction kinetics becomes an important aspect in designing the thermal process parameters for a food. Thank you.